So our two 60-80 ratios, sample 645 and sample 428 were in between the 1.5 to 2.2 ratio, which means that we isolated pure DNA, or there's pure DNA indication. Sample 427 was 2.39, which means that it was above the ratio, and this result would affect the rest of our results, so forth. 1.26 for sample 365, meaning that our DNA was in fact contaminated. So this UV spectrophotometry determines the concentration of DNA and predicts the contaminating proteins, as I just exhibited. So then we went on to gel electrophoresis. This is a technique used to separate DNA, RNA, and proteins. We use this technique to separate DNA fragments of different sizes in order to discover if our DNA isolation was successful. Our attempt to separate DNA was unsuccessful, and as our positive and negative control samples were the only ones provided us with data. So this is the ladder on the side right here, which ranges from 100 base pairs to 1,000 base pairs. And if you can see this little light up part right here, it is the positive control, and it registered in about 550 base pairs. The negative control and all of our samples did not register at all. The negative control was not supposed to register. So the positive control and the negative control were successful, but all of our samples that did not register were unsuccessful and indicated that we isolated no DNA at all. So this is a more elementary view of this, which is a picture of the actual gel electrophoresis that we did. So then we use the Ampere XB kit to clean up amplicon from our PCR reactions. This is done so that old used primers must be removed. Old primers are removed through the use of magnetic beads, which bind to complete double-stranded DNA, not single-strand primers. We then added short sequence tags to later identify our samples by using an indexing PCR reaction. When we sequence the DNA, we'll be able to identify each sequence using the indexing tags. So this is just some PCR calculations that were taken during the lab, and it shows like all the samples and the volume that we needed to use of the master mix and the water, etc. This shows the fluorimetry, which was taken from the positive and negative controls samples, which is why they were the only two that we could do. But then, because none of ours worked, we selected new reads from sample data online. We selected 54P, 54N, 7D344, and 1E 429. 344 was a husky and 429 was a corn snake. So the total reads range from 9,234 to 26, but they're all 100% of the reads passed and qualified for quality filtering. Then this data was taken and further analyzed by kingdom level, order level, and genus level. And order level is only split into unclassified kingdom level is only split into unclassified and bacteria present. So as you can see, the colors indicate which type, 7D, 1E, 54N, 54P, what type of if there was bacteria, and what type is just unclassified. For these, it's a little more in depth, and percent of reads is on the y-axis, and the type of bacteria is on the x-axis, and there are over 20 types of bacteria in all, different bacteria for order level and genus level. And the highest reads were in Endobacteriales and Escherichia. So there's an unclassified category on all three of these graphs, and the unclassified category is basically because only the top 8 classifications of bacteria were shown in each graph and recorded in the tables. The remaining classifications were not specified, and they're recorded under unclassified. On the pie chart, which isn't shown, there is also another category, which is the sum of all classifications with less than a 3.5% abundance. So the method used to quantify DNA absorbance was UV spectrophotometry. This method's advantage is that it accesses the quality of DNA and determines protein contamination. 260-280 is indicative of the purity of samples, and the value of 1.8 is the optimal value for pure DNA. After extracting DNA from the fecal samples, higher levels of absorbance indicate greater concentration of DNA present within samples. Our results, as seen on the gel electrophoresis, did not come out well, and the amplicon PCR loaded into the wells migrated straight down. Positive control stopped at 600 base pairs, around 550, while the negative control has no isolated DNA and is mainly water and migrated all the way down with the rest of the PCR results. The absence of a PCR product band at approximately 550 base pairs tell you that there is a form of cross-contamination. For a group, the amplicon PCR results as seen in the gel electrophoresis were not visible and migrated down. This shows the contamination and lack of DNA. For our group results, we chose new animals to compare their gut microbiome, and they're a corn snake and a husky. Total reads for husky are 9,234. The total reads and all reads passed the quality filtering. 86.27% of the reads of the bacteria at kingdom level went through, while the corn snake 1,746 total reads passed quality filtering at 92.21% of the bacteria went at kingdom level. These numbers are adequate to base an analyzation off of. There will be a difference in the gut microbiomes of the animals we tested, and there was, as our hypothesis was correct. At the order levels, huskies mainly consist of Bacteroideales, Clostridiales, Fusobacteriales, while the corn snake order level consists of all of those, but instead of Fusobacteriales, it consists of Enterobacteriales. Blaussia or Blaussia obium is a certain species of anaerobic bacteria found in the gut and are in both species of the husky, corn snake, and other breed of dog. B. obium and other bacteria play an important role in the recovery process from cholera infection that is toxic to the body and causes intolerable symptoms. So, to conclude, our data was correct, or our data was not correct, so our hypothesis, based off our data, our hypothesis could not be concluded, but based off the sample data that we took online, our hypothesis was good and correct. The end.